I'm pleased to have Dr. Joanna Barber here with us today. Joanna obtained her Ph.D. in analytical chemistry from the University of Florida. She joined Caco-2 XenoTech in 2007 and currently serves as the Vice President of Scientific Operations. Dr. Barbara also serves as an adjunct graduate professor of pharmaceutical sciences for the College of Pharmacy at the University of Missouri, Kansas City. She has authored or co-authored numerous scientific posters and papers and has represented Caco-2 XenoTech as an invited speaker at various analytical and drug metabolism conferences. Joanna has extensive experience in DMPK, regulatory compliance, and process and project management. As Vice President of Scientific Operations, she is responsible for the development, design, operation, and improvement of Caco-2 XenoTech's scientific division. It is my pleasure to present to you Dr. Joanna Barbara.
Thank you, Mike. So today we're going to be talking a little bit about challenges and solutions in today's drug transporter research landscape. We've had some changes recently that have impacted how everybody's starting to do some things, and I thought it would be kind of fun to go through a few case studies focused on the way that we need to think about transporter research nowadays. So I'll start out with a very brief overview of transporters in drug development to get everybody on the same page who doesn't necessarily do this or see this every day. And we'll talk a little bit about the specific updates in the 2017 in vitro drug-drug interaction guidance. Again, only focusing on the transporter aspect this time. But the bulk of the time that we spend together today will be spent on looking at a few case studies with some interesting features and some solutions that apply to them, and the lessons that we've learned. The data that I will share are fake but based on real data, and any structures I show have been significantly modified, so they are not the true structure that was originally supplied, but the principle is the same. So transporter assays are pretty interesting, and the reason that they're interesting to me is as a analytical chemist and problem solver are that you can design them in multiple different ways to answer different questions. There's the very simple assays that happen in drug discovery. You know, is my drug permeable? Can it cross a cell membrane? All the way through to pretty complex kinetic things, kinetic assessments in really interesting assay formats. We'll touch on that a little bit later on. When the compounds do what they are supposed to do, things are pretty straightforward, and so you can often run them in a fairly routine design and get useful results. But the problem, as is the case with so many things that we do in drug development, is that when the drugs are not cooperative, understanding the data that you get and establishing a path to move forward and make some decisions based on the data is actually quite difficult. It's a funny paradigm as a scientist because those are the most interesting ones. Those are the ones that my team and I will sit around and talk about and work through, and we kind of enjoy it. But of course, there's a delay and a cost associated with that, and so we all have to be cognizant of what we're doing. There are a lot of human transporter proteins, and when we're talking hundreds. But fortunately, in the drug development space, we don't have to worry about all of them. In general, we're going to be focused on the big 9, previously the big 7, mostly centered around hepatocyte uptake and efflux, and intestinal uptake and efflux, and renal uptake and efflux. Sometimes we get a lot of questions about what is the best format to study a transporter interaction in, and it's a really good question, but it doesn't have a very satisfactory answer, unfortunately. The way that we are limited when we're looking at drug transporters is basically by the availability of the test system that we can use to look at the transporter. The most common ones that we work with are polarized transfected cell lines, cell-based systems in culture. Generally for us, it's Caco-2 or HEC293 or MDCK2, dog kidney cells. And we do as many things in cell-based format as we have transfected cells available. The only way that you can study a transporter is to have a test system where you have that expressed in such a way that you can do a useful assay in it. We also do some work in hepatocytes, although a little bit less. That's more useful for, efflux uptake kind of work. And then when we don't have a more complete test system, we'll move to membrane vesicles. And those are useful, but they're not our preferred approach unless there isn't a different option available. It's much like a reaction phenotyping study. You'll use the recombinant CYP enzyme if you have it, but it's not a complete test system and there will be skewed results. So you have to be cognizant of that when you go into the assay that you're working on. Each test system needs its own set of conditions and controls to give you realistic and useful data. And so there is some limit in terms of chemical space in how you can design the assay itself. I'm going to move forward to talk about the 2017 DDI guidance a little bit. When this came out, which was about September last year, it revolutionized a few things in the ways that we do work here, and of course other CROs and big pharma, small pharma, all had to start looking at the guidance and figuring out, okay, what do we already do? What do we need to change up? How can we make our assays meet these requirements that now are rather more prescriptive in the FDA guidance than they used to be? Several changes were made to transporter assays. So the way that we did this, the way that we reacted to the guidance at XenoTech was we basically set up almost a response team, and it was composed of my brilliant colleague Brian Ogilvie and I as the core members, and then we had various specialists involved in helping with each discipline that was touched upon: reaction phenotyping, induction, inhibition, and transporter. And Andrea Wolf, who is one of our directors here, was very heavily involved in addressing the changes associated with transporters. So we as a team worked through the guidance, and these are some things that we found. I'm not going to cover in detail all of the changes because my colleague Brian and also our founder, Dr. Andrew Parkinson, have put out some pretty nice webinars on this topic already, where they really truly do detailed analysis of DDI guidance changes, not just for transporters, and also a comparison with the PMDA. So if you're looking for that, that's a really good resource. It's on our website. But what I'm focused on is the more practical aspect of how we dealt with accommodating the changes on the transporter side. Some of these were very easy to accommodate. One of the more prescriptive suggestions in this guidance was upping the pre-incubation for OATP assays to 30 minutes. Now, our assay already had a pre-incubation, so it isn't too difficult to say, guys, just don't do anything for 15 more minutes. That's an easy one to accommodate. We can handle that. Some are a bit more challenging. There is a discussion about needing 2 inhibitors for transporters in certain of the assay formats. That's tough because this is still an area of rapid growing and learning, and there are not 2 specific inhibitors for every transporter that are known and reproducible and reliable. So you have to be a little bit creative in terms of how you deal with that. But some of them spoke to very practical considerations and what you do with the data and what it means. And that's something that I find very interesting, so that's what we're going to talk about today. One of the areas that we saw a big change was that the FDA specifically called out certain considerations that people have been making in these designs for a long time, but not necessarily considering as essentials. To us, as practitioners of these assays, they absolutely make sense. There's this list here of specific issues to address, and in fact there are several others that I didn't cover, but the big ones that do make a difference are stability and non-specific binding. Solubility limits and cytotoxicity are more focused on what concentrations you can practically test as opposed to concentrations you might want to test because of the guidance recommendations. But all of the other things are very practical nature. And why is that important? Is because if you don't consider these things and the data are not straightforward, it's very difficult to make a decision on what you're going to do next. So it absolutely made sense to us, but it meant that we had to address some things in terms of our normal standard offering and incorporate sharing this information as part of our process. So the first case I'm going to speak to is focused on solute carrier proteins. The actual study was substrate and inhibition potential of a drug for these various solute carrier proteins. The client in this case had already got some basic data, had already got some data on P-gp and BCRP, and this happens quite a lot, is a client will have some early data or partial data, and they'll come to us and they'll go, what else do I need for my submission? And then we can do a gap analysis and go, you're missing this, here's a consideration, here are things you have to do, here are recommendations, etc. And that's kind of a fun part of the job, is getting to do that with a client. But in this case, it was fairly straightforward because they already had some of it and they clearly didn't have any of the rest of it. So we just spent some time working through the solute carrier proteins. And I'm only going to focus on the inhibition side of this study because that was the most interesting area in terms of the topic of today's conversation. So one of the very important proteins that we have to look at is OATP1B1. It has a partner 1B3, and so almost everybody has to look at those. They're hepatic proteins. They're very important in drug disposition, uptake and disposition. And these are the kind of data that we routinely see. I'm going to spend a moment just talking about what you're looking at because everybody presents data differently. So this top graph right here is our standard 1B1 inhibition graph. This assay format is pretty simple. Well, not when you have to do it, but when you're looking at the data, it's pretty simple. It's an OATP1B1 transfected cell line and then a control cell line that isn't transfected. The gray bars are the transfected cells and the white bars are the control cells. And you incubate a probe substrate and then you add your drug and see if the increasing concentrations of the drug change the uptake of the probe substrate. So gray bar being big and white bar being little is just saying transport associated with the OATP1B1 protein. And then as the gray bar decreases, while the drug compound increases, it means that we're inhibiting the uptake of the probe substrate because this is all data for the probe substrate. So this is a pretty classic inhibition response that we would see in this assay. And when we go ahead and plot the data and calculate an IC50, we get a lovely IC50 of 11.2. The positive control data look great. We have solvent control, so no inhibitor, no drug, gives us really nice uptake, clearly related to the transporter protein, and then in the presence of the inhibitors, we drop all that active transport down. So that's a pretty classic 1B1 inhibition data set. In this case, we saw a very similar trend in 1B3. When I first started doing this, I had sort of assumed that in general, 1B1 and 1B3, you would either inhibit both or neither. It's not strictly accurate. But in this particular case, it did hold true. Again, we see a decrease of the gray bar as we increase concentration of the drug, but in this case, we get a lower IC50 of 6.2. And of course, the lower the IC50, the more problematic things are. Positive control data look great again. For the sake of comparison and completeness, I wanted to show just a picture of what a negative result looks like, so everybody understands there's no room for confusion here. This is OCT2 data, and there's no inhibition at all. The gray bar just stays flat, essentially flat all the way through. There is biological variability, and so that's why it's a little bit bouncy, but it definitely tells you the story of no inhibition. And again, the positive control data look good, so you know it's not a problem with the assay. Now I'm going to sidestep for a minute and talk about what we do next with the data. The first round way to deal with the data when you have them, the IC50 data when you have them, is often to look at the FDA's basic model and compare certain parameters, certain calculations with the prescribed cutoffs in the guidance document. It's fairly straightforward math, but sometimes getting hold of all of the variables is quite difficult because it all depends on how much work people have already done, and so sometimes we have to use assumptions. The most interesting one for us is this top one, the 1B1, 1B3 equation. This recommends in our value, if these things multiply out to be bigger than or equal to 1.1, then there's recommendation for further follow-up work. And what they are is just the fraction that's floating around that's free that the transporter is actually experiencing, the concentration and the inlet, the portal vein, and then you're setting that into context with the IC50 value. The other ones are a bit simpler because we're not talking about, you don't have to worry about the inlet so much. You're more focused, particularly for PGP, BCRP, it's just IGUT, concentration in the gut over IC50, is it greater than or equal to 10? And then these two are Imax unbound instead. It's funny in transporter land because a lot of the things that in metabolism land would mean a thing have a different name. To me, this is a lot like Cmax, and so that's how I put it in context. But the point is, you can get all these parameters and then compare them to the cutoff values and make decisions based on that, no matter what you call the variable. Cmax, Imax, it doesn't matter. It's the same thing. The challenge for this compound is here. When we did the recovery assessments that now we have to do based on the FDA guidance, and often, to be honest, we were already doing because it is such an important variable to think about, we saw that there was a problem. We typically will assess recovery in the absence and the presence of cells because we want to know what we've lost, what material we've lost in the absence of cells, i.e. adsorbed to the assay apparatus, and we want to know what might be stuck in the cell monolayer. So that's our standard approach. And in this case, with no cells, the recovery isn't great, but it's not too bad. We're looking at a sort of real panic number is about 70%. There's various pieces that play into it, variability and looking at the data set and things. But in general, if it's above 70, we can usually make pretty solid conclusions. But in this case, as you can really see at the high concentration, which is unusual, we had a really nasty problem in the presence of cells where we actually lost half of the material. And we know, because it's different to the apparatus recovery, we know that it's lost to the cells themselves. So the first thing we did was go ahead and perform the calculations. We have our values for the 1B1, 1B3 pair. And what we found when we did the math was that this 6.2 IC50, compared to the concentration data and the dose and things that the client provided, the client already had them, resulted in an R value of 1.17, which is, of course, bigger than 1.1, which means it has potential for clinically relevant inhibition. Now, that doesn't necessarily mean that you're going to jump straight to a clinical study and you'll have to. What it means is some more attention has to be paid, perhaps a little bit more complex modeling and things to find out your real problem. But it's pretty important, and everybody wants to know that before they're already in the clinic, because then it gets to be expensive. However, as we talked about just now, we technically have lost about half the material in some of these measurements. So what that means is that the theoretical IC50 may not truly be the experimental IC50. It may not really be what the transporter protein is actually experiencing. So based on timing and the way where the client was with this drug, what they needed, various other pieces of information they had, we talked about all of the options of what to do next because there are multiple. And we decided that the best option for this one was the very simplest approach of figuring out what is the worst case scenario. If we truly have lost about 50% of this material and the IC50 is really, instead of 6.2, 3.1, what does that mean? So we went back through and we did all the calculations, assuming the worst case scenario. And in this case, the NSB corrected IC50 values also now bring the R value for OATP1B1 to above the 1.1 cutoff. And so when we're looking at this conservative scenario, it tells us that we need to consider 1B1 and 1B3. Well, at this stage of the game, designing a study, a clinical study, if you have to go there for 1B1 and 1B3 is pretty trivial. You're already looking at 1B3 anyway. But if you've already done the 1B3 study, that's a much nastier, much more expensive problem. So it's pretty important to take into account the literal pieces of the assay you're practically doing in order to understand your data and make appropriate decisions. Case 2 is a little bit different. In this particular one, we were looking at P-gp substrate potential, and we were looking in Caco-2 cells. We have multiple formats for P-gp, but the most common one that we work with is Caco-2. This is an interesting assay because it's performed in the transwell format, which is a little bit complicated. And it's a polarized cell monolayer, that's how it works, but the apparatus that are involved are interesting, and the way that you set up the assay are interesting. And for people that don't have direct experience with this, I wanted to take a minute just to explain some of the basics. This part here is essentially a well, and then there's a tiny little insert that sits in the well, just like any permeability, really. And then it has a little support for a membrane, and the cell monolayer sits right here. The basolateral side is the big side, and the apical side is the little side in this case. And so when you put drug in this side, the basolateral side, and you then find some in the apical side, because of the way that the cells are cultured and polarized, that means that there's active transport. Conversely, if we see movement from the apical to the basal side, that's passive permeability. We typically like to do bidirectional assays because it gives us better data, and it's just more reproducible, but sometimes people will only do unidirectional. In that case, all they care about is the beta A. One of the interesting things about this is that because of the way the assay is set up, the insert only has a volume for us of about 200 microliters. That's what we do, whereas the actual well itself has a volume of about 1 ml. And for us, when we do this work, the donor side is just wherever we put the drug. In a unidirectional assay, it would be basolateral, but for us, donor is wherever we put it, receiver is the other side. The reason that the volume matters is because technically the drug is experiencing a different environment on either side. Because we have this low volume, the surface area to volume ratio is much higher in the insert on the A side than it is on the basolateral side in the rest of the well, and that becomes important later. So when we're doing this assay, sometimes we forget to go back to the basics. What are we actually doing? What are the principles? What is this assay based on? And the ultimate results are based on some pretty old math, actually. The permeability is generally based on Fick's first law, which is very old, where he was talking about intestinal permeability. And what he proposed was that the flux across the cells was equal to some permeability coefficient and directly proportional to the intestinal concentration experienced by the cells. Pretty straightforward. When you go to a transwell assay, it's not quite as straightforward because you have to be thinking about the donor and receiver, but the principle is the same. Now what we use for comparison is PAP values, these guys. So we're looking for the apparent permeability coefficient. And the actual equation itself is fairly straightforward. It's just volume of the receiver corrected for the surface area of the membrane with a factor of the donor concentration at time 0, whatever you just put in there, and then you multiply it all by the flux. In this case, flux is very literal, meaning how much changes with time. Pretty simple. But this is an interesting one for those who like math, because these units are the same, right? So they cancel out, which means you can technically replace it with different parameters as long as they're the same. And so the equation works just as well for mass as it does for concentration, and that's usually the way that we do things, because we want to have mass balance. If we've lost some drug, we want to know these things. So for us, this is an important and useful feature. It's the exact same equation, except now I'm looking at amount in moles and change in amount in moles instead of the concentration. That becomes important later on. So transwell assays particularly can suffer from non-specific binding problems. All of these assays can, but transwell assays have the particular problem because of the surface area change, surface area to volume ratio change that I mentioned earlier. What you have to bear in mind here is that the compound can bind to the apparatus, it can bind to the cell monolayer, it can bind on this side, it can bind on the inside. And so you really need to be thinking about that, because for substrate assays in particular, you have to be able to measure to very low levels. Even if just a little bit has crossed from the donor side into the receiver side, you need to know that. It's very rare that none crosses the membrane, even in situations where there isn't real transport. So we need to be able to measure the compound all the way down to nanomolar levels, which is challenging enough, to be honest, in the salty buffers that we have to use for these test systems. But also, if you've lost half the compound to non-specific binding, now you're at sub-nanomolar, and it can get quite difficult. So here's an example of some of the recovery data for this kind of assay. I'm showing these data because it's kind of a fun one, because we had 2 time points. These data are very old. And it used to be that for a particular client, we would look at what the experimental concentration was compared to the theoretical of the stock before we do anything with it. And then in the absence of cells, we would do a 2-minute incubation and find out how much of the drug is still there. And we would do a 90-minute incubation because that was the longest time point for the assay itself. And in this particular case, the recovery at 2 minutes is pretty good. It ranges from 78 to 96%. But at 90 minutes, the recovery is not so good anymore. This is more classic non-specific binding data. You generally see a bigger effect at the lower concentration because the relative amounts are much more relevant to a smaller concentration than a larger concentration. So this is classic situation. At 30 micromolar, we have pretty nice recovery, almost 90, but as you go down in terms of concentration, you end up all the way down to 42% recovery. So in this case, in the absence of cells, we've lost almost 60% just through adsorption to the apparatus. Whereas in the presence of cells, we lose a bit more. Now, these, again, are the old data with the long time point. We have learned from doing this for many years now that actually it's diminishing returns after about 20 to 30 minutes. And so nowadays we would do one time point around 20 or 30 minutes depending on some things. And then we would use that value in the control cells and the apparatus to correct for everything, or just to have in your back pocket depending on what we want to do. But in this case, we're doing the 90-minute. For the transwell assay, you truly have to measure recovery apical to basal and basal to apical because they are very different, as would be expected. But it's important in terms of a consideration to make because you have to know what you're doing. You have to know what the data mean when you get them. And it can be quite confusing in terms of understanding what you have at the end of the assay. So in this case, they're all lower. The basal to apical is kind of okay, we could work with that, but the apical to basal is much, much worse because of the high surface area to volume ratio. We've lost a lot to the control cells anyway, we lost a lot to the apparatus anyway, sorry, we just saw that, and now we've lost some to the control cells as well. So we know at this point that some is trapped in the monolayer. So here is the example of the PGP substrate data. These are PAP and efflux ratios calculated using the normal equations that we talked about, but they're a little bit depressing. This is a situation, this is probably the most depressing situation that I see typically is when I see something like this, these data for 0.3 micromolar drug. So per the FDA guidance, in order to say that a drug is a substrate, you need a net flux or efflux ratio of greater than or equal to 2 in an appropriate test system, and it needs to be inhibited by one known inhibitor at a concentration taking into account sensible kinetics. The problem with any cutoff is what do you do when you're right at the cutoff? Because technically this is smaller than 2, but it's essentially 2, and it appears to be decreased by the presence of the inhibitor. So for us, that's almost inconclusive. And then the same thing happens at the higher concentration, the 3 micromolar. It's not data that makes you very confident. However, we are not taking into account the non-specific binding in this situation yet. And we can. There's a really nice paper in Drug Discovery Today, it's very old, it's from 2003, that explains what to do and how to fix this problem. And it's a nice paper because it's literally called something like old forgotten tricks that we already knew that need to be applied today in terms of whatever assays we're doing. And for all of us, sometimes we will overcomplicate because of the newer technology and forget the basic principles of what we're doing. And it's an article that discusses that and talks about simple corrections and things. So if we go back and we look at the mass equation for the apparent permeability coefficient, we have this donor concentration. But really, truly, what we need to be thinking about when we're talking about flux is the donor mass, taking away the amount that we've lost in the monolayer, because it's trapped, it's not doing anything. And it's pretty simple to do it. It's not intuitive necessarily unless you think it through, but it's a pretty simple correction. And all you have to do is figure out how much of the material is trapped in the monolayer between time 0 and the time points. We usually have multiple time points and we will sum them all. There's another way that you can do it where you can just pick the biggest time point and use that as an assumption. We typically don't. We typically will sum them all. So that's what we did. And so when we correct the data, we get much easier to follow information. When we've accounted for the material lost in the monolayer, now we, at this 0.3 concentration, have an efflux ratio of 1, which is much more useful. It's pretty conclusive. It's clearly below 2, it's not at 2, it's not above 2, very straightforward. And now, because we've taken that into account, the presence of the inhibitor clearly doesn't make all that much effect. In fact, it doesn't make any difference in this case. And the same thing has happened with the 3 micromolar concentration. It wasn't as borderline, so it wasn't as worrying, but anytime you have a yes at one concentration and it's the lowest, and a no at another concentration and it's the highest, you're always a little bit worried that maybe you've saturated the transporter and the true answer is a yes. So it's much more encouraging to have a data set that looks like this. Now we have conclusive data that tells us, okay, it's not a PGP substrate in this assay under the conditions we've tested. We know what to do next. So when we take the M cells into account, it makes a big, big difference to whether or not we can use the data. Case 3 is my favorite. I enjoyed this one. This was really fun. It started out as a very simple Caco-2 permeability screen. In transwell format, it was 1 concentration with our PGP inhibitor valspadar, 1 time point. We did very, very quick and dirty LCMS method development because it's screening. And we were looking for the normal things that we look for, PAP, efflux ratio, recovery measurement. And we always include high and low permeability controls just to make sure we know what's going on. But the data were not very useful at all. So this is screening. We have minimal information going in. I don't know what the drug is. I literally am blind at this point. And what we found was that there was nothing in the receiver samples. I mentioned earlier it's very rare for there literally to be nothing in the receiver samples. Usually you'll see a little bit, because the idea that nothing crosses the membrane at all, it doesn't make practical sense. But in this case, there was literally nothing detectable in the receiver samples, which meant that we couldn't calculate any PAPs. The efflux ratio is the PAP B to A over the PAP A to B, so I can't calculate that either. So I essentially have no permeability information about this compound at all, but what I do have is recovery data. And the recovery data are very strange. Here we have almost 400% recovery. That clearly is wrong. Something is clearly wrong. And so I'm a bit ashamed of myself on this, but I actually talked to the analyst who did the assay, and I didn't believe that she had done it correctly, which was very unfair because she's a really good analyst and she does things correctly almost all the time. But it was tough for me to believe that everything had gone right. And so we did repeat the assay. I should have known better because all the positive control data look really, really good. Mitojoxin, which is expected substrate, expected PGP substrate, has a beautiful efflux ratio of 36.6. I have plenty of a nice high PAP when I have active transport happening and a low PAP when I don't. The low permeability control mannitol does exactly what you would expect, low PAPs, boring efflux ratio, recovery is nice, and the high permeability control does exactly what it's supposed to do. I have higher PAPs than the mannitol, but still efflux ratio is 1 because I have equal active and passive transport, basically. And the recovery is nice. The interesting piece here is that usually when you see nothing in the receiver samples, you kind of assume it's a non-specific binding problem. But the excessively high recovery would definitely speak against that. You wouldn't usually see higher recovery. In fact, you just wouldn't see high recovery because it would all have been bound to something else. So something interesting is happening. So the assay was repeated and the exact same results were obtained. The analyst had indeed done a very good job and done exactly what they needed to do. Those were just the data. So at this point, we stopped and we talked to the client because usually in a screening scenario, there is no troubleshooting. It didn't work here, or it did work here. So we had to have a conversation with the client as to whether or not they wanted to proceed to look into things, because it would take more time, it would take more money. We hadn't built that into the scope of the study, but the client did want to proceed and did want some answers. They needed the data. And so they obligingly shared the structure with us. And my background is metabolism and analytical chemistry. So as soon as I saw this drug, I went to look in amide, and we're in cells that metabolize. It's hydrolyzing. Probably 10 seconds of structure review. Now, I obviously didn't know that was true necessarily, but it's a pretty solid hypothesis. It's fairly simple. And if it did hydrolyze, then it should theoretically convert to two products. This thing with the free amine, and then the other side of that is the free carboxylic acid. So if the theory holds true, and there's enzymatic hydrolysis, because remember, these are cells, they're metabolically active, we have a long time point, then this guy should form and should have a really strong electrospray signal for LCMS. This guy would be tougher because it doesn't have anything interesting. This is actually, even behind the box, isn't real, but the point is that there's not an awful lot that ionizes well on this structure. Sometimes carboxylic acids will ionize nicely in negative. Sometimes you can get a sodium adduct in positive, but they're not easy to work with. In contrast, this guy is pretty easy to work with. So the first thing we did was explore if this made sense. And we got a nice product ion spectrum for the compound standard itself. And we were really, really lucky, because the way this fragments would give us really nice diagnostic fragments all on this side for the free amine product, product A. So we know that if we find it, we'll be able to tell pretty conclusively whether or not it's present. Once we had this for the standard, we looked at the donor samples. The donor samples do have a little bit in there, but not very much. So the next thing that we had to do was look in the donor samples to see if the hydrolysis product is there. We don't have a standard for it, it's a hypothesis. So the only place to look in it, much like in MedID, is to look in the actual sample. Is there a thing there? So we looked in there, and fortunately we found it. We have a nice accurate mass, but we also have, more importantly, the diagnostic fragments that tell us that the compound of this mass is associated with the drug and is the structure that we've proposed. So now we've confirmed that there's hydrolysis happening in these samples. We looked again, and we found product A was also detected in the receiver samples. That's very interesting, because it cannot have come from the drug itself on account of the fact that the drug itself isn't making it into the receiver samples and is only in the donor sample side. If we go back to the math and think about it carefully, we can make some pretty interesting conclusions. In the normal assay data processing, we talked about this, the units for these two variables are concentration. But if you think back to school and your dimensional analysis, the bottom line is all of these units cancel out. I have centimeters cubed up here, centimeters squared here. So when I divide the one by the other, I get centimeters. This is concentration, concentration, it cancels out. And this is delta T, so time in seconds. And so what I actually end up with truly is that all units cancel except centimeters per second. This is a relative measurement. The units of concentration don't matter. The units of mass don't matter as long as they're consistent here. Why is that important? Well, it's important because I have no way to find out what the concentration of the hydrolysis product is, but what I can do is find out the relative amount between the receiver and the donor sample, and I can then use that to calculate flux and the original substitute for concentration, which means that I can technically calculate the P-app using the area ratio of the hydrolysis product, even though we don't have a reference standard. Now, we've known this from before because we did some screening tests many years ago, because we could recognize from the math that this was a relative measurement, and really it ought to cancel out, but we've also proven with some internal data that it works, so we're fairly confident in it at this point. When we go back and do that, we find some pretty interesting things. In this case, what we realize is that the hydrolysis product would almost certainly coalute with the original drug for various reasons, mainly centered around the fact it was a short screening method because we weren't supposed to be having metabolism, we're looking at transport only. So we elongated the method because the fragment that we were looking for in the MRM transition actually was exactly the same as a hydrolysis product, because usually those amides will give us really nice fragmentation data. And that explained the ridiculously high recovery. Ionization efficiency is pretty high for a free amine in general, and so it looked like 400%, but really it wasn't. Really it was nothing of the drug essentially, and then whatever of the metabolite was floating around, giving us the one signal. So when we did that, we elongated the method, we ended up with sensible recovery. Now everything ranges between 76 and 100, much better. We still don't see anything here, which isn't a surprise because now we know that there basically isn't any drug left to cross the membrane anyway. In contrast, the amide hydrolysis product gives us beautiful data showing active transport. The P-app in the beta A active direction is 62, whereas in A to B passive direction is 26, giving us an efflux ratio of 2.37, much bigger than 2, and then it decreases in the presence of the inhibitor valspadar. So what we were able to do is confirm that the drug was unstable and converted to its hydrolysis metabolite in the incubation, and perhaps more intriguingly, that the hydrolysis product A was undergoing active transport across the Caco-2 cells. So in conclusion, overall, we've talked about why these factors highlighted in the 2017 guidance are very important, and we've really focused on non-specific binding and stability because those are the more interesting ones. But the important thing to take away is there's a really good reason these are highlighted. There are really good reasons to take these into account, and sometimes they're actually critical to whether or not you can generate useful data. When I'm trying to teach some of my newer people, I always like to say things about, what are you doing? Where was this? Where did it come from? Remember the practical factors of the assays and the assumptions that you're making when you're really doing this. What is the purpose of what you're doing? And if you think about all of those things, you can usually reach useful conclusions, even if at first glance it looks like it probably isn't going to give you much. And then I also have a plug. Please give partners your compound structure to access their knowledge, because if I had had the compound structure in the first place, this would have been a much simpler assay, and this does happen. We see compounds all the time. We have a lot of experience, a lot of knowledge on a lot of different things, and we can do the best job to help you guys if you share the information with us up front. I do have to acknowledge a few people, Dr. Ogilvie and Andrea, who both were very instrumental in working through our response to the DDI guidance and also in much of the troubleshooting that we've discussed today. And Rebecca and Lois were study directors for some of the studies that I've based the data on. Andy, who is the wonderful analyst who I forced to repeat this assay even though she did it right the first time. I think she deserves a shout out too. And then Chase McCoy, who did all of the hands-on practical work for case number 3, because I don't really go in the lab anymore these days.
All right, thank you, Joanna. Information about our transporter services, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Joanna's presentation or any of Caco-2 XenoTech's consulting and contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website. This wraps up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Dr. Barbara for her wonderful presentation. Have a great rest of your day.